So this is DNA. So this is a quick little cartoon just to kind of introduce the lesson to you. So as you can see from the model, the structure is basically a long, twisty rope laddery type thingy. Okay, so this was a illustration from 1953, which actually described the model of DNA. So what does DNA actually stand for? So it's deoxyribose N-nucleic acid. Okay, so deoxyribose nucleic acid. So what does it actually do for you? It codes for your genes or your traits, and it's made of repeated subunits called nucleotides. So what exactly is a nucleotide? It has to have three parts. You've got a phosphate group, or deoxyribose, which is a sugar, so anything that ends with ose is a sugar, and then you have four bases, A, T, G, and C. So you need to have a phosphate group, a sugar, and a base. So the nitrogenous bases are grouped into two different types. You've got your double ring structures, which are called your purines. These are A and G, so pure agony. Okay, or your single ring, which is pyrimidines. These are your T and your C. So pure agony, A, G. You can remember those. Pyrimidines are your T and your C. So base pairing. So purines only pair with pyrimidines. There's 3 hydrogen bonds, okay, that are required between guanine and cytosine. So if you can see in the illustration, so you have 3 hydrogen bonds between the two. So there's 2 hydrogen bonds that are required between adenine and thymine. So here's a quick little question for you. So if there's 30% of adenine, how much cytosine is present? So the answer, there would be 20% cytosine. So if you think adenine, there's 30, so A matches with T, so there's 30% of thymine. Guanine is 20%, so there must be 20% of cytosine. Therefore, 60% is A and T's, 40% is G and C's. So a double helix. So it kind of looks like a ladder, if you will, or a straightened ladder. So you have the legs of the ladder. This is your phosphate group as well as your sugar backbone. You have your rungs or your steps of your ladder, and that's your bases, your A, your T, your G's, and your C's. So the base pairing rule. Adenine always, always, always binds with thymine. Guanine always binds with cytosine. So again, the sides of the DNA are the phosphate group and your sugar, and they're held together by hydrogen bonds. So do you remember if hydrogen bonds are really weak or really strong? So let's do a little bit of a practice. So you have your base pair rules. Okay, so remember, A's go with T's and C's go with G's. So you have the adenine would bind with thymine. Thymine would bind with adenine. Adenine would bind with thymine. Thymine would bind with adenine. Cytosine would bind with guanine. Adenine would bind with thymine. Thymine would bind with adenine. Guanine would bind with cytosine. Cytosine would bind with guanine. And our last 3, guanine, 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 would bind with cytosine, cytosine. So this is a quick little video of what's the relationship between your genes and your DNA that I want you to take a look at. So how are your DNA, what's the combination? So you have these 4 base pairs, but what actually determines your traits? So if you look, it's your coding, or the combination between your A's, your T's, your G's, and your C's actually determines what traits you have. So for example, for this purple people eater, if they had CAT, that would be coding for purple hair. But if you had TAC, that would code for yellow hair. So think of bases as basically like letters. Letters form words and words form sentences. So you have an endless combination between your base pair roles. So let's do a little review. Okay, so DNA stands for deoxyribose nucleic acid. What is the shape of DNA? That is a double helix. So who established the structure of DNA? So there were 2 gentlemen. It was Watson and Crick. Okay, and the last one, number 4, adenine always pairs with thymine. So the sides of the DNA ladder are deoxyribose and the phosphate group. So guanine always pairs with cytosine. So what is the complementary sequence, or the sequence that matches? So if we have an original DNA strand, which is CAT-TAG, what is its complementary, or what it's going to match to? So you should have G-T-A-A-T-C. The two sides of DNA are held together by what kind of bonds? They're hydrogen bonds. And are they very weak or very strong? They're very weak. So DNA is composed of repeating subunits. Remember those 3 parts that they have to have? You have to have a base, you have to have a sugar, and you have to have a phosphate group. What is that repeating subunit called? A nucleotide. So what are the 4 bases? It's adenine, thymine, guanine, and cytosine. Let's talk about our next statement. So, or next process, I should say. So DNA replication. So DNA has to be copied before it divides. So do you remember the 2 different processes that when cells divide? Mitosis and meiosis. Okay, so DNA is copied during the S phase, right, of interphase. So is this the longest phase or the shortest phase? It's the longest phase. And new cells will need identical strands of DNA. And so the first phase is the S phase. Okay, so this does happen during interphase. It is the longest. It occurs in the nucleus of eukaryotic cells, as well as if we look at our cell cycle. Okay, so we have G1. Remember, G1 is for growth. S is where the DNA replicates. G2 is where it preps for mitosis. So mitosis, you have 4 phases. You have prophase, meta, ana, tele, and then last is cytokinesis. So replication, okay, so basically it begins at the origin. There's 2 different strands that formed, okay, it's called a fork, okay, it looks like a Y-shaped region. So the new strands grow from the forks. So they grow from either side. So basically you have a 2-way system that you're actually replicating. So when we look, again, we have 2 DNA strands that open at the origin. We call them replication bubbles. Okay, so with bacteria only has a single bubble. Eukaryotic cells can have many bubbles. So if you look, you can have many parts or many sequences that you are actually replicating at once. So the idea was presented by Watson and Crick. Okay, so remember, Watson and Crick were the 2 gentlemen, the 2 scientists that actually discovered the model of DNA. So we they refer to this model as semi-conservative. So basically what that means is that each of the 2 strands of the parent molecule will separate. One will act as a template and one will act as a complementary strand. So the new DNA is, again, 1 parent or 1 original plus 1 new. Okay, so if we look, this is just an illustration to show you exactly how the replication fork, aka the replication bubble, starts. So again, 1 is the new, okay, and 1 is the old. So if we look, the lighter gray on either side of the strand, this is actually the original, and the darker gray in the middle, those are the new strands, or the new of the semi-conservative strand. So again, there's 2 enzymes. Remember, enzymes speed up chemical reactions? Well, these enzymes are extremely important. We have to know the difference between helicase. Okay, so anytime you see ACE is a enzyme. Okay, so helicase unwinds or unzips the 2 strands. Okay, because again, it can break those weak hydrogen bonds. So the second enzyme that you need to know is polymerase. Polymerase attaches new nucleotides to create that new strand. So basically what it kind of looks like is it kind of looks like the claw machine at an arcade. So it's going to grab new additional things. So proofreading. So polymerase basically can initially make 1 in 10,000 base pair errors. Why would that be a bad thing? Well, if you don't have enzymes to proofread and correct these mistakes, what's going to eventually happen to the organism? Well, what happens is you can have mutations, okay? So there's a new error rate for DNA that has been proofreading 1 in 1 billion base pair errors. So I want you to take a couple minutes and look at this video and to state and to show you how the helicase versus polymerase works. So sequencing, okay, so we refer to this as central dogma. So you go from DNA language to RNA language to proteins. So proteins, again, are the building blocks of most organisms. So RNA is ribonucleic acid, okay? So again, ribose is that sugar, but this is the messenger. So some difference between RNA and DNA is it's single-stranded. It has a ribose sugar. It does not have thymine, so there's no T's. But there is the new base, which is called uracil. So again, it follows the exact same base pair rules, but instead there's no T. Okay, so A matches with U and vice versa. So if we have this DNA strand of A, T, A, C, G, C, G, what would our RNA strand be? It would be U, A, U, C, G, C. So if you look, you'll notice again, this is the messenger, and it's going to carry the message to the ribosomes, which again, ribosomes are there where proteins are made. So it's single-stranded. So again, what's so important about these proteins? Remember that proteins are essential for life, okay? So you have carrier proteins that help move things in and out of the cell, okay, of the cell membrane, like endocytosis. You have recognition proteins, which again identify themselves, okay, according to the immune system. You have antibodies. Remember, antibodies are those that actually help defend and disable viruses and bacteria. You have structural proteins, like the ones that are in your ligaments and tendons that are connection tissue. And then you have hormones, which hormones deliver messages throughout the entire body. So the enzymes, again, anything that ends in ACE, it's going to speed up a chemical reaction. So let's look at insulin. Okay, so insulin is for it's for your blood sugar. Okay, so proteins are made of basically one or more polypeptides. So poly means what? Many, and peptide forms chains of amino acids. Okay, remember what amino acids are? It's the basic unit of a protein. Okay, so the insulin, which is a protein hormone, again, this regulates your blood sugar. So it's made of many peptide bonds. Okay, so proteins are pretty interesting because, again, based on their structure, determines their function. So you have primary structure, which is the first sequence. So even a slight change, a slight mistake between one amino acid can actually distort the entire shape, and it can make the entire protein unable to function. So let's look at transcription versus translation. Okay, so transcription is where you go from DNA language to RNA language. Now, translation is where you go from RNA language to protein language. Okay, so let's start with transcription first. So just like those important helicase and polymerase, DNA polymerase, you have with transcription, you have again another very special enzyme which is called RNA polymerase. So what it does, just like DNA polymerase, it binds to a specific gene or a specific DNA sequence. Okay, so this new molecule is actually called mRNA or messenger RNA, and it's basically going to use DNA as a template language. Okay, so again, the nitrogenous bases are red, okay, just like RNA, or just like DNA polymerase, it's red, just RNA reads it just the same. But what it does is it reads a unit, okay, which is called a codon. So each codon translates into one amino acid. So the first codon, or the first starter, is called a start codon. Sometimes we call this a promoter, okay, so it is AUG. Okay, there's also a stop or a terminator codon, which basically it reads the message all the way until it hits the stop. The stop can be UAA, UGA, and UAG. So how I remember it is University of Georgia, UGA, UGA, UAG, and UAA. So the messenger RNA basically is going to generate the new or the required polypeptide. So basically this is translation. So translation is going to take each of those codons and it's going to match up with an anticodon. So anticodon are 3 bases that are found on tRNA. Okay, so for example, if you have the messenger RNA, which is CCG, the anti or the match is going to be GGC. So the tRNA carries a specific amino acid, which is going to create a new chain or a new chain for that. So if you notice on the graph here, you have to, if I have a C, 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 purple, 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 so I'm going to go from my first position to the left, my second position, which is up top, and then my third to the right. Where they all match up is the particular amino acid. So for CCC, it's pro for proline. Okay, and you'll notice that those UAA and UGA and UAG are all for coding for stop. Now remember, AUG is for met, or it's for your starter. Okay, but you'll notice that some of them are non-ambiguous, which means that basically they are specific for one amino acid. So you might have a codon, like if you look down at the bottom for Val, you have 4 different codons that match for Val. So keep that in mind. So this is a short little clip to show you translation versus transcription. So I want you to go and look on your phone or look this up when you're at home. Okay, so some animations for replication versus transcription versus translation. Okay, so I want you to be able to label this image. So you have DNA. Okay, so DNA is inside the nucleus. Messenger RNA has the ability to move from the nucleus out, okay, through the pores. You have your anticodon, which is number 3. Anticodon is associated with the T of tRNA, so anti-tRNA, and it's associated with one particular amino acid, which kind of looks like a ball or a head on the tRNA. So the codon is only responsible or only associated with mRNA. So you have these ribosomes, which are sisters through this transcription and translation. And then of course, once the amino acids bind together, you form one big protein. So let's look at this illustration and let's label. So number 1 is your what? Your cell membrane. Number 2 is where all of the organelles live. That's your cytoplasm. What's number 3? It's inside number 4. So number 3 is your DNA. What is inside? Where's your DNA housed? Number 4 is your nucleus. Number 5 is where what process is occurring. Okay, so it's going from DNA language to RNA language. So number 5 is transcribing. Okay, you're going to transcribe it or write it in a new language from DNA to RNA. What's number 6 that can actually leave out of the nucleus? That's your messenger RNA. 7 is a portal or a hole, which is called your what? Your nuclear membrane or nuclear pore. Okay, number 8, what is that unit, that device? That is your tRNA. Number 9, that is where all of these things are being attached and it's going to grow into one big chain. That's called your polypeptide chain, or what your proteins are going to be. What's number 10? Where you go from mRNA language to a protein language. That's translation. Okay, so what number 10 also can be is what is the organelle that assists or makes protein. That's your ribosome. And number 11 is your messenger. So again, we talked about insulin. Okay, so remember, insulin is a protein it's a protein hormone that regulates your sugar. Okay, so how exactly is insulin built? So the double helix of chromosome 11 is unzipped by DNA helicase. Okay, your mRNA is transcribed, okay, from your DNA strand. So the mRNA leaves the nucleus, okay, and it's going to go down to the rough ER because it has to reach a ribosome. Okay, so the mRNA is going to be coded for a particular amino acid. So is that transcription or translation? When you go from mRNA language to protein language, that's translation. So basically what's it going to do? It's going to hit that stop codon, and then it's going to disassemble, and all of the protein is built. Once the protein is built, the insulin can go to the Golgi, which it's packaged and it's shipped out through the cell membrane. Okay, so let's talk about mutations. So we've talked about mutations when we talked about genetics. So a mutation is anything that is a change in DNA sequence. So you have substitution, which basically that means that the wrong nucleotide was inserted. Okay, so adenine, or excuse me, a thymine was inserted instead of an adenine. Okay, so an insertion means that you just have an additional nucleotide. A deletion is it's just completely skipped and it's not added. So let's look at substitution mutations. So there's some that they're called silent mutations that don't really have an effect on the structure of the polypeptide or the structure of the protein. So a missense results in basically an insertion of one single mistake, okay, one single incorrect amino acid. So sickle cell anemia, this is actually a result of a missense. So one tiny little mistake actually caused the hemoglobin protein, which is in your red blood cells, to change and mutate. So a nonsense mutation means that there's a change in the codon to a stop. So it's going to prematurely stop or terminate the assembly of your protein. Okay, this is the most damaging kind of substitution mutation. So a frame shift, this can be either an insertion or deletion. We call them frame shift mutations because basically it disrupts the reading, okay, or the reading group. So if you look, each of the codons code for one particular amino acid. So if it goes from GLU to now LSY, okay, just because of that one change between the guanine to the adenine, okay, it's going to disrupt the entire reading sequence for the rest of the gene. Okay, a mutagen. Okay, a mutagen basically causes some kind of, this could be either a physical or a chemical agent. Okay, this actually causes mutation. So you might see this warning sign. This includes UV light, UV rays, X-rays, asbestos, and echo. Okay, so let's look at the history. So early scientists actually thought that proteins were the factor or the material that actually you gained from your parents. Because again, proteins are actually composed of 20 different amino acids in these long, huge chains. Well, what happened is we had a scientist, Griffith, that used two different strands of pneumonia. Okay, so you had R strands and S strands. So he noticed that the R strands could be fatal. Okay, but what he did is he actually took the DNA from heat killed. Okay, so he basically took the bacteria and he put it under a fire. So he heat killed the S strand. So he noticed that because of this, it was suggested that DNA was probably the genetic material and it wasn't the protein themselves. Okay, so this is what actually he took. So what he did is he took the R strand and the S strand, he killed the S strand, and when he injected it into the mouse, he noticed that the mouse was dead, even though it was killed, the heat killed. So let's go back to basic genetics. Okay, so chromosomes are made of both DNA and RNA. We know that. Okay, so Hershey and Chase actually wanted to do a spin-off on Griffith's experiment. So what they used is they used a bacteriophage. So a bacteriophage kind of looks like the lunar spaceship, and it is a virus. Okay, that basically only attacks bacteria. Okay, so they wanted to prove that DNA was actually the genetic material. So what they did is they used radioactive phosphorus that was injected into the bacteria. Okay, so we use basically glow-in-the-dark phosphorus that injected. Okay, And what they noticed is they noticed that the phosphorus actually was injected into the bacteria, and the bacteria made new radioactive phosphorus. Okay, so our next scientist was Cherkov. Okay, and Cherkov was the one that showed the amounts of adenine, thymine, guanine, and cytidine. So in a body or a somatic cell, we know that there is 30% or 30.3% of adenine. So again, there's 30.3% of thymine. There's only 19.5% of guanine, but there's 19.9% of cytosine. Okay, so he knew that adenine must pair with thymine, guanine must pair with cytosine. So, and those bases are formed with weak hydrogen bonds. Okay, so your purines and your pyrimidines. So our last lady, who's probably the most important out of all of them, okay, Rosalind Franklin. So what she did is she took X-rays of DNA. So this is an actual picture of her X-rays. So, and what she found out is that it was an actual double helix. Well, whoa. I thought that Watson and Crick were the first to build the DNA model. Well, what they did is they actually used Franklin's X-rays to solidify or confirm their structure. Okay, so they used her data in order to figure out that it was a double helix. Well, here's the catch-22. So back then in the 30s, so our last one that we're going to talk about is the Human Genome Project. We talked a little bit about this when we talked about genetics. So the Human Genome Project was actually an international research effort to basically figure out or map and sequence all of our genes, which is called a genome. Okay, so for our species of Homo sapien. So it started in 1984, but it wasn't completed until 2003. So if you notice, that's almost 20 years of research that multiple companies and multiple countries actually were working together as a collaborative effort in order to sequence this. So basically, for the very first time in 2003, we were actually able to complete the entire genetic blueprint for us as human beings. So we use this data and this information to come up with new drugs and treatment for medical research, and also for commercial uses, and just for that's how we know for Ancestry.com or Chromosome 23. These companies in these areas are able to use the Human Genome Project to help create or help identify these different disorders.